Okay, welcome back to A-level biology revision. So, what I want to do today is I want to look at the 2019 paper. I don't think we've gone over this one yet, and we're gonna look over paper 2, which of course we have in just a couple of days now. Oh, it looks like we've already gone over this one. Okay, that's fine. I will go over 2018. Hopefully I haven't gone over that one before. Okay. This is a fresh paper. Gonna rub off all of this witchcraft stuff because yeah, we have that exam on Thursday, but biology is more important. Okay. So, let's get on with this. First question. Okay, photosynthesis question. Heat stress is a condition that often occurs in plants exposed to high temperatures for a prolonged period of time. Heat stress is a major factor in limiting the rate of photosynthesis. Heat stress decreases the light-dependent reaction. Explain why this leads to a decrease in the light-independent reaction. Okay, this is a lovely question. It's the product of LDR. The product of LDR is ATP. Products are reduced NADP or NADPH. And what's the other product? There's another product, what's it called? Oh, I don't remember. There's another product of a light-dependent reaction. Water? But I don't think that matters anyways. Anyways, the two products we focus on is ATP and NADPH. ATP is used in the light-independent reaction, I think, to reduce triose phosphate into GP? No. Oh, why can't I remember? Hold on, Krebs cycle. It's RUBP plus CO2 catalyzed by Rubisco gets us, is it GP and then TP? I think that's right. That goes off to become carbon. 5 carbon goes to be RUBP. Okay. Massive long-winded explanation here. ATP, I believe, if memory serves correctly, ATP is what's used, the energy released from ATP is used in part to reduce 2GP into 2TP. So, less/no ATP, less/no reduced NADP. Cool, okay. 2 marks, nice. I'll keep a little tally. We'll keep count. So yeah, the two products of a light-dependent reaction, ATP and NADP. Obviously, I've just explained the role of those in this particular process. The Krebs cycle, the Calvin cycle is really nice, I think. There's not a lot to remember compared to the Krebs cycle, where there's a lot of different things going on. Another 2 marks. Another effect of heat stress is a decrease in the activity of the enzyme Rubisco. Okay. A decrease in the activity of an enzyme means the rate of reaction it catalyzes becomes slower. A decrease in the activity of Rubisco would limit the rate of photosynthesis. Explain why. Okay, This is perfect. This is just what I've explained. Rubisco catalyzes the reaction between RUBP plus CO2 into 2GP. Now, I'm going to assume that that's only worth a mark, and I'm going to assume that the second mark point, because it's an enzyme question, I'm going to assume it wants you to mention that less enzyme substrate complexes will form. Just a wild guess though. Let's see. Less/no oh, okay, no, I didn't have to put that, which is surprising. Less/no carbon dioxide reacts with RUBP, less and no GP. Okay, I think I would have got that in an exam. Less RUBP reacts with CO2. Yes, that's what I said. Less GP, because GP is the product. Okay, cool. Wow. Those were two lovely questions with barely any mark points. Like, wow. Where precisely... Oh, shit. Where precisely is rubisco found in a cell? Is it the stroma in a... We draw a chloroplast. Thylakoid discs. The lamellae connecting them. Whereabouts are they located? Is it here? Because I know that the thylakoid membrane is where the LDR happens. Is the LIDR the stroma? No idea, just a guess. Yes, it's a stroma, okay. Wow, I'm bigging myself up here because I'm actually surprised I got that right. Reject stoma, reject stroma, reject stroma of mitochondrion, ignore references to Calvin cycle. Cool. 5 marks. That is a godsend. I would be praying for a question like that. 5 marks to start off, and it's about that. Like, that's lovely. 5 marks to warm you up. Scientists investigated the effect of temperature on the activity of 2 enzymes, rubisco and rubisco activase. Okay. We've been given an unknown enzyme. I will wager you money that it's going to tell us about the enzyme substrate complex in the mark scheme. The scientists concluded that heat stress reduces the activity of rubisco in plant leaves by affecting rubisco activase. Evaluate the conclusion. Okay. If it's an evaluation question, you want to look for agree and disagree. Usually you'll get a mark point for talking about a statistical test. Or, if not that, you'll get a mark point for talking about samples. So let's look at this. We have a graph here. And a graph here. So this is rubisco activity at different temperatures. And it only goes up to 50 degrees. That's something to note. Meanwhile, rubisco activase activity is a bell curve. And it drops off after about 60 degrees. Like so. So. Already I'm noticing a flaw with this graph. This only goes up to 50, this only goes up to 60. Heat stress reduces the activity of rubisco by affecting rubisco activase. I can't see that. I can't see that from this diagram, from these graphs. This is rubisco activity. This is rubisco... Oh, wow, I see I've done that wrong. That's rubisco activase. I don't see that. I see that the temperature causes rubisco activase to stop working, but it doesn't affect Rubisco. So can we disagree here? Can we say that at 50 degrees, at 50 degrees, Rubisco activity remains high, we'll say at 9. Comparatively, Rubisco activate activity drops off after about 30 degrees. You could then say there is no fall off in Rubisco activity after 30 degrees, which would suggest heat stress does not affect Rubisco. But you could counter that by saying that this chart only goes up to 50, this only goes up to 60. So you could argue that in Rubisco, the optimum temperature is going to be higher. Rubisco activity increases with temperature, or Rubisco optimum temperature is above, yes. Rubisco activate activity decreases at high temperatures above 25 degrees. That's what we said, 2 marks. Results suggest activate does not affect activity, 1 mark. And I dropped a mark. Because no stats test. I told you. You always get a mark for saying no stats test. I didn't vocally say it, but I think in an exam I might have said it. Also, Mr. Mark Point, a very obvious one. Same species. They're only testing one species. So how can you confirm that it's for all Rubisco if it's only being tested in one species? I'll happily drop a mark for that, because that was a fun question. That was an evaluation question. So remember, the holy trinity of evaluation questions. No stats test. If they haven't done a stats test, you can't confirm if the data is statistically significant. Number 2, only done it on one species, or done it on a small sample size, so not representative of population. And 3 is usually some kind of in-between point. Like, you could say that, conditions might not reflect environmental. Anyways, out of 9 marks, we got 8. Very happy with that. That is a very good start to that paper. Out of 91 marks, we've almost already got 10%. Okay. There are different types of gene mutation. Put a tick in the box next to the statement which describes incorrectly the effect of a mutation in an exon, so a coding gene, of a gene. Okay. Get rid of this. My only advice for paper 2, really, that I haven't said, is literally just be descriptive. Be descriptive. You could lose marks just for forgetting the tiniest thing. Like, I forgot to say a couple of days ago that myelination is insulation, and I lost a mark because of that. So literally, if you think something, say it. Even if you don't think it's worth a mark, just write it down. Because what do you have to lose? The mark schemes are very picky. Something you think might not be worth anything could be worth a lot of marks. Anyways, there are different types of gene mutation. Right, which statement is false? A substitution may not result in a change to the encoded amino acid. Okay, a substitution mutation, that's where if we have our base sequence of, let's just say, CAT, AAG, you are substituting a gene, you are replacing one. Now, key thing to remember with substitutions is it does not lead to a frame shift. Now, a substitution may not result in a change to the amino acid. Well, yeah, that's true, because DNA is degenerate. What degenerate means is that multiple codons will code for the same amino acid. For example, CAT and CTT could code for I don't even remember amino acid names, like glycine? That's not true, I'm just using that as an example. But yeah, DNA is degenerate. So many base sequences will code for the exact same, even if it's slightly different. An inversion will result in a change in the number of DNA bases. That's false. If we have CATAAG, an inversion mutation is literally just going to flip that. Literally just flips it. That's all it does. It doesn't add, it doesn't remove. So the number of bases remains exactly the same. So that is the outlier. Let's go over the others though. A deletion will result in a frame shift. Yes, it will. CATAAG. If we remove the A, you see that we are now missing a codon. Same with addition. CAATAG. That is obviously going to lead to a different number of base sequences. So those are the mutations. Inversion, substitution, these are the two that lead to a frame shift, aka changing the number of bases / codons. These two don't change the number of bases and codons. So, yes, an inversion. Okay, box 2. Describe how alterations to tumor suppressor gene... Oh my god, I can't speak. Describe how alterations to tumor suppressor genes can lead to the development of tumors. Alterations, that implies methylation, because methylation... Oh shit, which one is it? Hypo-methylation and hyper-methylation. I don't remember which one is which. Okay, maybe this is something that we need to revise then. So methylation can either lead to the histones becoming tighter or looser. What that means is that, what does it mean? What does methylation do? That will either promote the coding of oncogenes, or it will inhibit proto-oncogenes. Let's have a look. Increased methylation of tumor suppressor genes. So 1 mark, yeah. Mutation in tumor suppressor genes. Tumor suppressor genes are not transcribed or expressed. Results in rapid cell division. Okay, let's learn that. Accept abnormal methylation or hypermethylation. Ignore decreased acetylation. Okay, so literally, hypermethylation. This is something that I do not remember. Just 1 sec. Hypermethylation. So increased methylation of tumor suppressor genes leads to mutation in tumor suppressor genes. Tumor suppressor genes are not transcribed / expressed, which results in rapid and uncontrollable cell division. Okay. So I'm assuming hypomethylation is where... Hold on, let's actually Google this. Let's Google this, because I don't remember. Hypermethylation It does not say. It literally does not tell me. Can I get a diagram, maybe? Hyper leads to the activation of genes. Hyper silences genes, preventing them from doing their function. Hyper promotes uncontrolled cell growth because genes are activated. So, hypermethylation leads to tumor suppressor genes not being transcribed or expressed. I don't know why you wouldn't get a mark for saying, I don't know why you wouldn't get a mark for saying silenced. I feel like that would be a nice mark. I'll add that myself to my document. Increased methylation of tumor suppressor genes. Tumor suppressor genes are not expressed, results in rapid cell division. That's a nice one. Okay, I get it now. I understand. So methylation, hypermethylation, which is the main one you want to focus on, silences genes because the increased methylation leads to tumor suppressor cells not being expressed. Okay, so I would have gotten a mark, I would have dropped 2. Next question. A type of malignant tumor, where's my calculator? Type of malignant tumor divides every 8 hours. So that's 3 divisions a day. Starting with one of these cells, so 1 times 2 to the power of something, how many will be present after 4 weeks? Okay, so this is 1 times 2, 2 to the power of 4 times 7 times 3. Right? Which is 2 to the power of 48, no? 21, 42, 84. 2 to the power of 84. I believe that that's the answer. Correct answer of 2 times 10 to the power of 25. No, why would it be that? Wait, no, I'm being stupid, that's why. I don't think I've answered it properly. One times 2 to the power of 84 gets us, yes, 1.9. Yes, okay, we got it right. I got it right. Thank God, because that was a lovely question for 2 marks. Okay, anaerobic respiration question. Fun. Yeast cells can respire aerobically or anaerobically. A student used the apparatus shown in figure 3 to measure the rate of respiration in yeast. Okay, so it is literally just a flask like so, with a bung to obviously block air from entering. Then this is connected up to a glass tubing like so. And in it we have the liquid, the coloured liquid. Then we have a mysterious solution which absorbs oxygen. Then we have the yeast culture in a tiny little tube in glucose. So glucose will act as the respiratory substrate. Suggest one reason why it was important the student left the apparatus for 1 hour after they reached a constant temperature. So he positioned the flask in a water bath so the yeast culture reached a constant temperature. He then left the apparatus for an hour before starting the investigation. Is that just to give the yeast time to begin respiring? I don't know. I'm guessing it's to give the yeast time to begin to respire. So the oxygen is used / respired. Okay, yes. Oh, of course. So, they are still in aerobic conditions when the experiment begins. So giving it an hour allows all remaining oxygen to be absorbed. Okay, I didn't think about that, to be honest. But yes, that's why it's here. That's why it's here to absorb oxygen. It's there to absorb oxygen because there's still some there. If they wanted to create anaerobic conditions, they would add something like oil to the top. But it's still aerobic, so I lost the mark, but that's a very obvious point. During her investigation, the liquid moved right. So it moved from the left to the right. It moved this way. Why? Why would it move this way? Well, we are going to want to talk about how the yeast is going to produce CO2 and ethanol because it's respiring anaerobically. So there is no O2 uptake. So the only thing that's happening to the gas is there is more gas. More gas. Therefore, it's increasing the pressure due to the greater volume of gas release. Therefore, 2 marks. Anaerobic respiration produces CO2, increased pressure and volume. Correct. Okay, 2 marks. Oh, here's a big mark question. This doesn't look like it's a very lengthy... Oh no, there's more. I was going to say, this is like an oddly small anaerobic practical question. The student found that the coloured liquid moved 1.5 centimetres in 24 hours. The diameter of the lumen, so the hole of the capillary tubing, the diameter, so not the radius, was 1 millimetre. So the radius is going to be 0.5, just as a reference. And we have to calculate it in centimetres cubed per hour minus 1. Okay. So firstly, we are going to want to... how do we start this? The volume is given by pi squared times L. The volume of a capillary tubing is given by pi squared times L. Wait, I'm so confused. What does it mean by length? Right, let's do the most obvious thing first. 1.5 is the volume of gas. Let's divide that by 24. We get ourselves 0.0625 centimeters cubed. the diameter was 1 millimeter, so we could convert that. Let's convert it this way. 6.25 millimeters cubed. That's how much gas was produced per hour. I don't know where you would go from there. Incorrect answer, but shows the number 49. Incorrect answer, but shows a number from 1175 to 1178. Incorrect answer, but shows division by 24. Okay, I'm confused how you would do this. Is it literally just pi squared times length? But what's the length? How the fuck do you do this? I'm so confused. The student form of a coloured liquid moved 1.5 centimeters. Is that the length? Let's try this. Pi times 1.5 squared. No, sorry. Pi squared times 1.5. So we get 14.8. That is supposedly the volume of the tubing. Now it's saying that it moved, the lumen of a hole was, oh, I'm so confused. The correct answer is 49 times 10 to the power of minus 4. Do I divide this by 24? No, that gives me 0.6168. What the fuck? I have no idea how to do this. That is one of the first times a math question that simple for 2 marks has genuinely stumped me. And you fuckers were saying that the math questions on the paper 1 were bad. Like, no. This is an example of a bad math question where it's confusing as fuck. Explain why a log scale is used to record the number of cells. We know this, it's exponential growth. Accept references to exponential increase. So yeah. You could have also just said a large difference in numbers. Many yeast cells die during the death phase. So just one reason why. It's going to be the increase in ethanol concentration. Increase in ethanol / CO2 / acidity. Yes, you also could have said decrease in glucose substrate. So, the suggest questions are normally very nice. You can normally be very creative with them. Okay, how many have we got so far? 8, 9, 10, 11, 12, 13, 14, 15, 16. 16, and we've dropped 1, 2, 3, 4, 5, 6. So far we have 16 out of... We have 72% correct so far. I'm fairly happy with that. That would be an A star. Right, the final one I will do... Oh, there's still questions. The following equation can be used to make predictions. Oh, this is like a mad formula. Okay. Maybe I'll send this one to Declan. xt equals XO E RT. And it says... XT is a population after a certain amount of time. XO is the starting population times by the base of natural logarithm 2.72 times R growth rate and time. So, how do we apply this? A population of 2,000 yeast cells was left for 10 hours. So 2,000 yeast cells times 2.72 to the power of 10 times 0.5. So to the power of 5. Assuming no cells died, okay, let's calculate this. 2,000 times 2.72 to the power of 5 gets us 297,766. Correct. Yes, 2 marks. Nice. That was a funky question. Right, we will do, because this is, I'm assuming 10 marks, well 10 questions. We'll get up to 5, we'll finish 5, I'll get halfway through 5, then we'll go, because I'm tired. And it's almost 8, so I probably should get going in a minute. Scientists investigated the process of succession. Okay, succession is a lovely topic. They measured the percentage cover of different species of plants on sand dunes of different ages. Some results... Okay, I'm not drawing this graph, that is way too complicated. Describe how you would determine the mean percentage cover. Lovely question. This is a lovely question. This is using quadrats. This is a lovely question. I just need to remember how to answer it. Okay, mark point 1 is going to be talking about using random sampling. One of the mark points is going to be using a large sample so that you can calculate a average / mean that represents whole population or whole environment. So use random sampling. Use random sampling to generate coordinates. Then you would obviously place quadrats 1 meter by 1 meter on the coordinates and count the number of beach grass. You would make sure to use a large sample to calculate an average / mean that represents a whole population. Then you would multiply the average number of grass by the area of the dune. For 3 marks, we should have got all 3, surely. Method of randomly determining position. So, random numbers / table generator. Large number / sample of quadrats. If a specified number, divide total percentage by number of quadrats / sample / reading. Okay, I didn't put that. I said multiply. So actually, I only got 2 marks for that lovely question, which sucks. But okay, so the total percentage. So if it's like 23% of quadrats have them in, by number of quadrats / samples. So 23% divided by, let's say we took 20 samples, that's going to be roughly, how many is there? Oh, because it says percentage cover. Aha, I was trying to determine the number of individuals. I didn't read the question fully. Oh, the scientists concluded that the results shown in figure 5 were due to succession. Use figure 5 to explain why they reached the conclusion. Okay, so the first species that grows is beach grass. So what were you going to say is... Oh, shit. My bad. What we are going to say is beach grass is the... What's it called? It's not the founding species. Oh my god, what's it called? Why can't I remember? Oh my god, why can't I remember? What's it called? C... S... Starting species? No. Successor... Primary successor? Successor? No. What's it called? Oh my god, why don't I know? Founding species? That doesn't sound right. Successor? That doesn't sound right. It's literally, I can visualize this word. Founding species. It's like the word you would use to describe, like, Einstein or, like, fucking Galileo. I don't know what the word is. Why don't I remember? I'm gonna go insane. What's it called? I'm gonna kill myself when I find out. No. What's it called? What is the word for the first species? The first species. Colony. It colonizes. Algae is an example. No, not algae. Lichen on rock. Oh my god, why don't I remember? What the fuck is it called? Oh my god. Something species. S or F? What? No, I don't want to cheat. I will remember this. When the founding species dies, it creates nutrients that allow other species to grow and live. It's not the founding species. Okay, I'm cheating. What is it? Pioneer! Fuck! Oh, come on! I knew that. Oh, okay. You know what? I'm not even going to dock myself a mark because I fought valiantly there to remember and I couldn't. Pioneer. It was on the... I could literally visualise the word. But I couldn't, I just forgot. Pioneer. Pioneer. Beachgrass is the pioneer species. When they die, their death, the beachgrass death, after about day like 25, you want to say? After their death, it leads to the growth of other species. So after the beachgrass's decline in population due to their death, it leads to other species, it leads to the growth of other species. Then, finally, it reaches a climax community. So let's have a look at what it says. Pioneers change the abiotic conditions so it's more suitable for them species. Then the final species is climax. Okay, I need to drill this in my head. Succession, because I spent yesterday writing up a mark scheme for it, and I forgot. So, Pioneer changes the abiotic conditions so that it is more suitable for other species. Which eventually reaches a climax community. I am keeping that on my board because that is a really obvious one that I missed. Pioneer species changes the abiotic environment, making it suitable for other species. Okay. So that would have been 4 marks. I probably would have gotten 1. I can't lie. Or maybe 2. The reason I dropped so many there is because I forgot all of the basic critical thinking. Use figure 5 to explain the results in figure 6. Percentage of full sunlight reaching the ground drops off after 500. The reason why is because after 500, that's when you have the population of hardwood trees is growing. And it's increasing at a steady rate. And the trees block the light. So yeah, trees block and reduce light. Using evidence from 6, what can you conclude about the net primary productivity in sand dunes that are older than 1,000 years? Well, due to the fact that the percentage cover is probably around like 25%, you are going to want to say that there is less photosynthesis happening on the ground versus the trees. It remains constant. Photosynthesis is low. Or less light, so less photosynthesis. Okay, I have 1 mark. I'll happily take 1 mark. Fluorosemide and CBT are drugs used to remove excess fluid from the body. Scientists investigated okay, this might be an evaluation question. The effects of these drugs on the volume of urine produced by human volunteers. They divided the volunteers into 3 groups, A, B, and C. So, that's an important point. They divided them randomly. They gave them all the same food for 3 days. After 3 days, they gave the volunteers in A a tablet containing fluorosemide. The volunteers in B a tablet containing CBT, and C a placebo. So C is a control group. Some of the results the scientists obtained are below. Okay, so number 1, the first thing I'm noticing is the standard deviations do not overlap. That would suggest the data is statistically significant. Or, which one is standard deviation overlapping? That means the data was less likely to be obtained by chance, right? All the volunteers were given the same food for 3 days. Suggest and explain one reason why. It acts as a control, so that the diets they had before engaging in the experiment do not affect the results. Affects water potential, affects volume of urine. Oh, I would not have gotten any marks then. Okay. I hate that, actually, because I thought that question was asking me like a experiment question, not a science question. Suggest and explain one reason why they were given the same food. It affects water potential. Using table 1, what can you conclude about the effectiveness? Oh, there's no evaluation. What can you conclude about the effectiveness? Well, you can conclude that fluorosemide and CBT remove fluid due to the increase in urine compared to the control group. You could say that it's almost 4 to 5 times higher. You could also then possibly argue that the stats test. Yes, correct reference to significant increase / significant difference because they don't overlap. Okay, so yeah, statistically significant. And they're more effective than placebo, yes. Fluorosemide inhibits the absorption of sodium and chloride ions in the filtrate produced in the nephrons. Suggest how that causes an increase in the volume. Okay, well, this is in the loop of Henle. Obviously, they would be actively transported out, so we're going to say no. No absorption of Na plus / Cl ions. Therefore, that is going to lead to a lower water potential in the nephron. Therefore, more water will move in. What the hell? I just wrote mate. More water will move into the nephron via osmosis, which will overall increase the volume of the urine. Water potential decreased, less water reabsorbed by osmosis, and 1 mark for saying collection duct. I would have got 2 marks then. I talked about the loop of Henle, which apparently it was not on about. Two marks, though. The mean rate of flow of blood plasma is 60%. How much greater is the flow of blood in the kidneys with furosemide than with placebo? So furosemide is, don't mind me, furosemide is 380. Mean rate of blood flow into the kidneys, so it's 60%. So we multiply that. Divided by the placebo of 295 times 0.6. Right. Oh wait, but that's per minute. So then we also have to multiply that by 60 times 4. Why is that 1 mark? 34680. Can we confirm if that's correct? Yeah, go on. 380 times 0.6 minus 295 times 0.6. 51 times 60 times 4. No, I got it wrong. How much greater is the flow with furosemide? Right, let's try this a different way. I'm going to say 380 times 60 times 4 times 0.6. No! I don't get that. Wow. Accept answers in the range. I would not have got that. Apparently the math questions on this paper suck balls. Final question I'll do. In genetic crosses, the observed phenotype obtained in the offspring are often not the same as expected ratios. Suggest why. Fertilization / fusion of gametes is random, and crossing over occurs. No, wait. The observed ratios in the offspring. Accept crossing over / okay, yes, I would have got the marks. Fusion of gametes is random. Oh, and sex linkage, that would have been a good one. You also could have just said epistasis. That's all I'm going to do, because those questions then begin to delve into genetic crosses and all of that horrible stuff. I only have 1 lesson tomorrow, I believe, so that means I have ample time to revise for biology and history. I'm now going to go work some more on history, because unfortunately that exam is worth a third of my grades and I kind of need to do well in it. So yeah, ciao for now.